right, thank you and good morning. And as Lee mentioned, I'll be talking mainly about the genomic selection here at the Land Institute. Okay, so today, basically just cover a little bit about genomic selection and then some of the things once we have the program in place for genomic selection, what we can do with some of that data, looking at the genetic architecture of these traits, how is spike yield controlled, and what that means going forward within the breeding strategy. So just as a quick schematic overview, when we're thinking genomic selection, our breeding program, we have our selected parents that we want to intermate and create the next generation. As soon as we start those seedlings, then we can tissue sample that, extract DNA, sequence that, and predict how we think that plant is going to perform in the field 1 year, 2 years later, even when it's only a 5 to 6 week old seedling. So from that prediction, we can go ahead and select right away what we want to be or what we think are the best parents that we want to intermate again for the next generation. So this gets us down to a 1 year breeding cycle and timeline. If we think from the breeder's equation telling us how much genetic gain we can get, our time to complete that cycle is 1 year. So then our equation just gets to a function of how intense is our selection, how accurate is our selection, and the amount of genetic variation that's in that population. Now there is a second part. We want our GS model, our mathematical model that's predicting how these plants are going to perform. We want to keep that updated because as our population is moving, we need a model that's updating along with those plants. So in this case, the way we keep our model updated is we take plants that weren't selected in the best 100 parents, put them in the field, and then the subsequent year or 2 years, they can be phenotyped, and that phenotypic data then is used to update the model. So in this case, our genomic selection model is actually 1 year behind, or 1 year behind our selected parents. So data coming in from cycle 11 will be used to select cycle 12 parents. And just as a timeline, the way this looks in practice, I've got on across the x-axis the year starting in 2015, and then on the bottom axis the various cycles. And TLI cycle 6 was our first training population. It was the last cycle of just phenotypic selection. So it was planted in 2015 and then evaluated in 2016 and 17, at which point the best parents were selected out of that. Well, starting in 2017, we started genomic selection. We were able to select those best parents out, and then in 2018 start cycle 8. But at the same time, with a perennial plant, instead of evaluating just for 1 or 2 years, we've been able to maintain that evaluation out in cycle 7, up to 4 years. Last year was the last year of data collection. With cycle 8, we'll have another year. Already, even though we're evaluating these cycles for multiple years, we're able to cross that data. And one of the questions this morning, as far as persistence, getting that 4th year of cycle data, of yield data, allows us to retrain our model and look long-term without actually taking 4 years per cycle to get that information. And just for comparison, if we were to continue phenotypic selection at as fast as we could go, only evaluating plants for 1 year, right now in 2022, we would just be starting a cycle 9. So we've actually gained 3 full generations or cycles of selection by using genomic selection. And again, looking long term at these plants, we also get multiple cycles and site years of data, so that no longer are we constrained to having just evaluating cycle 6. Maybe it's a drought year, maybe it's a very wet year, and then the following year you're evaluating a different cycle under different environmental conditions. This gives us a little bit more robust model that we think is going to help increase our selection accuracy. So just looking at this, how has genomic selection been performing? This graph on the x-axis, this is our cycles going across from cycle 6 to cycle 11. The y-axis is the predicted spike yield for the 4,000 plants that are genotyped. And it ends up being over those 4 cycles, basically a 7% annual increase in spike yield. Another big trait that's been mentioned, free threshing. So we want a higher free threshing. These are the same type graph with the cycles on the x-axis. So our free threshing's increasing at about 11% annual rate per year. So this data is very promising. It looks like we're moving in the right direction. And then when we already have all the mechanisms in place, we've got a phenotype population, we have a genotype population, then we can go in and start looking at the genetic architecture. What are the genomic regions controlling these traits? So we can use a genome-wide association analysis, just using our breeding data that we've already accumulated to try to learn more about these traits and see if there's any particular areas of the genome that we should be looking at. So this was ran using the cycle 6 through 9 data. We looked at about 4,500 plants and about 22,000 markers. So one of the big traits is shattering. So we don't want plants that are shattering. And this is the Manhattan plot. Across the x-axis are the different chromosomes of the intermediate wheatgrass plant. And then on the y-axis is, you can think of the probability that region of the chromosome is influencing the trait of interest for shattering. So anywhere that there's points above the blue line, those are significant marker trait associations. Chromosome S2, S4, S5 all appear to be influencing shattering. And since we've got the entire breeding population, then we can go in and investigate these regions. So this is that chromosome S4, that one peak on S4. These are this is the marker that had that high significant association. And on the x-axis, these are different alleles for that single nucleotide polymorphism. So the homozygous C, the heterozygous, and then the homozygous G. And in this case, the shattering, the lower score is better. So we would like to see our population shift to having more C alleles than the G alleles. And then looking across those populations, how that gene has shifted due to selection. Starting out in cycle 6, you can see that the red reference G allele, that's the higher shattering, is actually more frequent in the population. And by cycle 9, the alternate allele, the C that has less shattering, is now the main allele in the population. And particularly we didn't select directly on this marker until we ran the GWAS. We really weren't even aware of this particular marker. This was all done with genomic selection, the black box mathematics, if you will. So it looks like it's pointing us in the right direction. So the last little bit that I'll cover is kind of how do we use this in our breeding strategy. This again is our same Manhattan plot that we were looking at with shattering. But on each of these peaks that are above the blue line, I've put in the amount of phenotypic variation that is described by each one of these markers. And often what we would like to see as a breeder is a major effect QTL that could describe 5 or 10% of the variation. And for this trait of shattering, we can see that most of these alleles are only explaining, 1.5 to 2% of the variation. So the GWAS, we looked at 34 different traits. Of those 34 different traits, only 16 of them could we find a marker trait association that was significant. There was no main effect or major effect QTLs. And even for known genes like brittle rachis, our largest effect QTL was only about 2.7% of the phenotypic variation, even though for a GWAS study, we had this large 2,000 to 4,000 individuals. We should have had high power to detect these things. And of course, everybody wants to know about spike yield, what associations are controlling spike yield. And, we didn't really find anything. But using genetic theory, we can go back in and estimate the number of QTLs that are controlling these traits. And this is a table kind of the major traits that we're looking at. And so the trait and then the QTL estimate, the minimum based on our sample size and power. And then using even a little bit more genetic theory, we can estimate an entire QTL range. So traits for like brittle rachis, at a minimum, we'd have at least 111 QTL, but it could be anywhere from 160 up to almost 300 QTL. And particularly brittle rachis, at least for the other annual crops, this is a well-known gene. It's well characterized. You solve the brittle rachis with BTR1, BTR2, and you've solved the problem. It does not appear that we have any major genes, at least that we've identified in our breeding population. So taking this, I like to think of it just that complex things are complex, but we can kind of manage this through clever designs. And one way that we can do this is genomic selection. It's a method it's been developed for these very complex situations to solve these answers. We know that there's few, if any, large effect QTLs, and we can adapt our breeding program going forward to take advantage of that information. So with that, I would like to acknowledge this work. There's been lots of individuals that have helped and responsible along the way, as well as lots of funding sources. I think if there's any time for questions, and thank you.
Yeah, we've got a little time. There's a question or two.
Do you think if if there are biparental crosses populations, the PV explained might go high, especially for the brittle rachis, because you have a huge population, as you mentioned, yet and very good hit, yet the explained percentage is low. That's what we see in Minnesota populations too, but I wonder if biparentals will help clarify it a little bit.
Yeah, so, and some of this work has been completed in the nested association mapping panel, but if we had a biparental where we had a known mother, father, I think we would probably get better trait resolution, and within that biparental family, we might see a higher percent variance explained, but then the challenge would be just like all of the annual crops, wheat, corn, where we have thousands of QTL studies, but in breeding practice, there's very few QTLs that have ever been used for marker assisted selection.